right, so this is okay. So this is where I'm working. Sorry, my phone is just ringing. It just started. I'm sitting right over here in this building right now, in Lookout, and it's a nice, pretty day, much like you see in the picture. So I will talk to you some about gene editing for infectious diseases, and it's great that Misaki talked about HIV because I don't have HIV data in this talk. We'll talk mostly about hepatitis B and herpes simplex. So I work at a cancer center, and so I often have to kind of justify why we work on viruses. And it's actually pretty easy because I think, as many of you may know, viruses are a major cause of human cancer. There's human papillomavirus, which causes a lot of cervical cancer, as well as head and neck cancer, which is increasing. Hepatitis B, which we'll talk about, causes liver cancer, as well as cirrhosis and other really bad pathology. You've already heard a lot about HIV, which is associated with an increased risk of Kaposi sarcoma, lymphoma, cervical cancer, and others. And then even herpes simplex, which itself isn't an oncogenic virus, but it does greatly increase the risk of HIV infection, about twofold, which means that literally millions of current HIV infections can be attributed to prior HSV infection. So all of these viruses actually contribute to cancer and are major causes of morbidity and mortality. So we would love to do things about them. And these are all persistent viral infections. That is, they typically have some sort of long-lived DNA form, and they go on for a very long time. And we have therapies now, and you've heard some of them in the discussion of the questions. So people, if they have access to good drug cocktails, can live relatively normal lifespans. With HIV, with hepatitis B, the therapies can reverse the liver damage, and people can do well. For HSV, suppressive acyclovir can reduce recurrences and the likelihood of transmission. It's not 100%, but it does reduce those. But really, the common theme here is that for all of these viruses, as soon as you stop therapy, the virus comes roaring back. So if a person stops their antiretrovirals, their HIV load will go up. Hepatitis B, the virus will return. Herpes, they'll have a recurrence. And the issue is, I mentioned that all these viruses have these long-lived DNA forms, and the drugs that we have now don't deal with that, which means they're always there. And if you have a garden and weeds in it, if you just keep plucking the top of the weeds, you never really get ahead of things. You're just keeping up. But wouldn't it be wonderful if we had a way to get down sort of into the roots and deal with what's allowing these weeds, the viruses in this case, to come up? And so here we are for HIV. It's integrated provirus that gets into the memory T cells. For hepatitis B, there's a thing called covalently closed circular DNA in hepatocytes. And for herpes simplex, the virus lives as episomal circles of DNA within the neurons. So what we would love to do is have a way to either eliminate the infected cell, or even better, get rid of the virus without the cell. And of course, in the context of this meeting that you're having, it's not surprising that the approach we've taken is gene editing. Now, I love the previous discussion about CRISPR-Cas and zinc fingers, because it's important to remember that gene editing is not only CRISPR-Cas. This is a wonderful way to gene edit, but it's not perfect in that it doesn't have every possible advantage. The huge advantage to CRISPR-Cas is that you can easily retarget it with RNA. It's RNA guided, which means that if I have a new target today, by tomorrow I can have a CRISPR-Cas attacking it. Things like zinc fingers are harder to engineer because it's actually a protein DNA interaction that's giving the specificity. There's other types as well. Tau effector nucleases had a short time in the sun. And then actually the first class is something called a homing endonuclease, or more commonly now called a meganuclease. And despite the name meganuclease, this is actually a very small protein. These are pretty much to scale. And so what you can see is this is very small, and that makes it a really nice thing for gene therapies because it's really easy to put into a gene therapy vector and deliver where you'd like it to go. The difficulty is that a homing endonuclease, a meganuclease, is very difficult to redesign, that is to give it a new specificity, because all of the recognition of the target as well as the DNA cleavage activity is within a very small single domain. And when you change specificity, you have to be sure you retain activity, and it's quite a challenge. So much more difficult. If I could have one of these for a new target tomorrow, I might have a new one of these in 6 months, and I wouldn't be sure I could do it. So they're very difficult, but once you have them, they do have some very good advantages. And I'm going to show you a little bit of data with both and sort of make the argument that for viral targets, the meganucleases may actually be the superior class of compounds. So the idea, both hepatitis B and herpes live as circles, again, either in the hepatocyte or the neuron. And what we're going to do is make cuts within that circular viral genome. And originally when we did this, we would target an essential gene with the idea that we'll target some essential gene in the virus. It'll ultimately get repaired, but there'll be an indel there, some sort of insertion and deletion. It will knock out that essential gene, and the virus will no longer be able to replicate. What we have subsequently found is that it's more efficient to actually make 2 cuts in the circle. And what happens is the cell has a very hard time finding these 2 separate pieces and actually bringing them back together to do the repair. What happens is this typically degrades. And so we actually get loss of viral genomes, and it turns out it's a much more effective way of eliminating virus. And it took us a couple of years actually to have that insight, and actually came from a work of ourselves and others in HIV, where there are repeats at the end, and we found if you targeted those, the whole virus would just come out. So that's what we'll do. We'll try to make double cuts in these circular viruses. Let's talk a little bit about herpes simplex virus. So herpes simplex infects at a mucosal surface, could be around the head and neck, could be around the genital region. And then the virus goes down the nerves that innervate that region, and it goes down to the nerve bodies and the ganglion that innervates that area. And that's where it establishes latency. Later on, it can come back to the surface, reinfect the cells again, and then amplify itself, cause a lesion, transmit, and all the other things. So we know where the virus establishes latency. It goes into this sleeping form where it lives for life in these ganglia. But this is actually a really nice target for a gene editing approach, because there aren't very many ganglia in the body, a couple dozen maybe. A ganglion doesn't have very many neurons, maybe 10,000. Not all of them get infected. And if a neuron does get infected, it doesn't have that many copies of HSV. So the latent viral load is actually substantially less than you might think of for something like HIV or especially hepatitis B. And we know that if you could reduce, or we think that if you could reduce the amount of virus here at the ganglion, you would reduce the likelihood and severity of what happens when the virus wakes up and tries to come back out. So it's actually a really nice target. So we had a collaboration with a French company, still do, for many years, who had developed a meganuclease, a series of meganucleases targeting herpes simplex. You'll hear me talk about some of them, one called M5, one called M8, and then another one I may mention in passing we've been working with lately called M4. And here's just the target. So they're always targeting an essential gene, but what we'll end up doing, just to cut a couple corners here and let you know now, we'll end up showing that it's more effective to use these in combination to make those 2 cuts that I talked about. We also have controls and so forth. And so we can start to think about gene editing the virus. And so, like many folks, we started, let's see, hold on, I'm going to try to hide that. Oh, there we go. So we wanted to do this just in a model system, and so let's see. Did I have? Yeah. So we did this in a culture system. So you can establish latent herpes infection actually in fibroblasts or some other cells if you do a lot of tweaking of the cells with interferon and acyclovir and temperature shifts and so forth. But But what we showed is that you could actually edit the episomal virus with a single meganuclease. And so if we look here, this is just the sequencing. It's back in the day, we did colony sequencing. It was no next generation sequencing or anything. You can see we edited about a little less than 10% of the latent virus in those cells. So we're happy with that. This was one of the very first, if not the very first, demonstrations of editing a latent virus, but 10% probably wasn't good enough to actually have clinical benefit. So we felt that to really turn this into a therapy, we needed to take it into animal models. And so we've done a lot of work in mice. And the basic model is this: you infect the mice with HSV, typically through the eye, and the mice get lesions around the eye, and those resolve after about 2 weeks, and everything calms down. So you give the mice about a month, and as far as you can tell, nothing has happened at this point. They're completely healed, but it turns out they have latent infection in those ganglia within the head. And so then we can give them some sort of therapy. What we typically do is use an adeno-associated virus vector to deliver the meganuclease. In this case, here's just the vector constructs. And then after 30 days, we harvest the ganglia and ask, have we had gene editing? And so we published this in 2016. And what you can see is in some of the animals, we absolutely did have gene editing in vivo now, in the ganglia, ranging from about 2 to 4%. So again, this was the first demonstration in vivo of editing of a latent viral infection. But again, 2 to 4% is probably not really enough to have any clinical benefit. So proof of principle, but not necessarily encouraging to ultimately have a therapy. Well, we kept working in trying to understand why things were low, like why the gene editing effect was low. And we went through a lot of different AAV serotypes. These adeno-associated virus vectors come in different types. And so they typically have a number or letters behind them. So AAV1 or AAV8 or AAVPHP.S and so forth. And so we actually studied how well do these go to the neurons where herpes lives? And this is an example of an experiment where we did a 10x single cell sequencing of neurons from infected and then AAV treated animals, and simply asked, if we look at where HSV is in green versus where the AAV went, or where they both went, what you could see is that there's different neuronal subsets sort of scattered in different areas throughout this XY t-SNE plot. But you can see that, for example, these are trigeminal ganglia, these are superior cervical ganglia. You can see the different AAV types have different tropisms for that, and the overlap is not really perfect. And so what we found is that certain AAV types could infect maybe up to 80% of the herpes infected cells, not all of them. But if we used combinations of these AAV serotypes, we could actually cover nearly all the AAVs, or excuse me, all the neurons. And so the triple therapy we settled on for quite some time was a AAV called RH10, AAV1, and then AAV8, and we could do those singly, in double pairs, or in the triple pair. And what we showed is that the triple pair was really, really good. So there's two big jumps we made to get to this point. One was using better AAVs and using them in combination. And the other one was that first data I showed you was with a single meganuclease to make that single cut that was repairing, and we were getting indels. But once we combined these good AAV serotypes together with the use of the dual cuts so that the circular episome would actually fall apart, what we saw now is, instead of looking at about 10% of just indel formation, we actually got 90% or more reduction in the viral load in the ganglia. So here's the controls. Up here, here's the viral load, how many HSV genomes per million cells are. You can see it's well over 10,000. And then after we treat with dual meganucleases, 2 meganucleases delivered by 3 AAVs, you can see that we get an over 90% reduction of HSV. So we published that in 2020 in this paper. We've subsequently, and this will hopefully be published soon, have evaluated this after genital infection of mice, again with HSV1. So you have to treat them with Depo-Provera, you inject HSV into the vagina, and then a month later treat them. And actually what we find there is incredibly good reductions. So here's the reduction within the dorsal root ganglia, and it's almost 2 logs. It's almost 99% reduction in those. And we believe this has to do with just how easily AAV can get to the neurons of the dorsal root ganglia. And it just has better accessibility to the blood for the AAV. So very, very effective thing here. And of course, one of the major concerns around HSV disease is genital disease. So we're very encouraged by that. Now, I know that kind of in the title of the workshop is CRISPR and Cas9. I will say we've worked a lot with CRISPR and Cas9 against HSV, and I'll only show you a little bit of data, but we've done this probably nearly 10 times with different collaborators. Cas9 really isn't very good against latent HSV. And here's just one experiment. Again, it's from the Nature Communications paper, but because we just wanted to compare a meganuclease head-to-head with Cas9. So we went through a lot of work in culture models to find good guides for HSV. So they're conserved, they have high activity in culture models. Everything looks great, and we can get probably 40% mutagenesis in the culture models. It's really remarkably good. But when we take those into mice, and what we'll use is AAV delivering Cas9 with 2 different guides, right? So we'll be able to make those 2 cuts, just like we talked about. What we find is that we never get a measurable reduction in viral genome. So this is a single meganuclease delivered with the same stuff. So you can see we don't get any reduction there either with that single cut. We weren't surprised by that. But even with 2 cuts with Cas9, we didn't get any reduction. And then if you look at the percentage of mutagenesis by next generation sequencing now, you can see that the meganuclease gives us nice measurable ones in many animals. And we see essentially nothing. So in some experiments, we can see vanishingly low but measurable levels there's a little bit here, for example, with this guide. But they're really, really low and not of meaningful value. So we have a meganuclease therapy where we can get 90 or 99% reduction, and a Cas9 therapy we can get almost nothing. And I'll tell you that we have worked with a lot of real experts in Cas9, and it hasn't worked well. I think it may have to do with the fact that HSV is heavily chromatinized when it's latent, and Cas9 I think really struggles with heavily chromatinized targets. It's chromatinized much more than most human genomic targets. So if you're targeting something in the human genome, it may not matter. If you're targeting a virus that's actively replicating, it may not matter. But when you're targeting a virus that's been repressed and is latent, CRISPR-Cas seems to struggle. So of course, if you could tell an infected human, for example, that we've reduced your ganglionic load by 90%, they might say, well, that's great, but what I really care about is, am I shedding virus? Could I infect someone else? Am I going to have lesions and so forth? And one of the problems with mice is that they don't spontaneously reactivate HSV. So they establish latency very nicely, but it doesn't come out spontaneously like you would see in a human. But we developed a small molecule model called JQ1. This is a bromodomain inhibitor that can actually wake the virus up and cause it to come back out. And we can just swab the eyes and measure it by a quantitative PCR. And then we can ask the question, if I reduce the ganglionic load, what do I do to viral shedding? And the answer is, if you reduce the ganglionic load, you have a dramatic reduction in shedding, and it's a little hard to follow through all these. But maybe we just look at this one. The control animals are shedding quite a bit of virus here, and then as we increase the amount of meganuclease, have more complete reduction in the ganglia, we have less frequent shedding at lower levels, and then at the highest level, we see absolutely none whatsoever. So you can actually, so the reduction of viral load translates into the sorts of things that human patients would care about, that is reducing shedding. Let's transfer now over to hepatitis B, another virus. Hepatitis B has some of the same problems as HSV. It also has some similarities to HIV. It has, for example, a reverse transcription phase to it. The virus infects, it goes down into the liver, Originally, it's kind of a partially double-stranded DNA form. When it gets in the liver, it fills itself out and makes something called covalently closed circular DNA. And this is what causes all the viral transcription. All the new viruses come from this. It is very long-lived and is continually replenished, so that if you wanted to cure HBV, you have to get rid of this CCC DNA form. And none of the drugs that we have now do that. They stop events out here, but since this lives a very long time, there are very, very few cures with current antivirals. So we're interested in gene editing for this, and here we don't have meganucleases for hepatitis B. We're working on that, but don't have them yet to show any data. So we worked with Cas9, not surprising. So we went through a series of screens for guides, and I won't go through all that, other than to say just the principles of what we're looking for here. We were looking for highly conserved regions of the virus, because we'd like this to work for as many people as possible. And as you know, hepatitis B has a lot of genetic diversity. We wanted the virus to, or excuse me, the guide to target a region that ideally would cover multiple open reading frames. Remember, hepatitis B has no wasted nucleotides in it. Every one is used for something. So it's a very compact virus, so you can cover multiple open reading frames with single cuts. And we just found in screening them that the guides C7 and C14 were the best in our screens. So we went forward with those. We did publish a paper showing that this all worked in the culture models of hepatitis B. Nick Weber was the first author on that. I won't show any data from that paper. But just like what I showed with herpes, we wanted to take that from culture experiments into animals. And that is a more difficult thing to do with hepatitis B. With herpes simplex, they're just kind of regular mice. You just infect them in the eye. There's nothing special about the mice. They cost about $10 each. So it's not a problem. For hepatitis B, this virus doesn't grow in mice. It doesn't grow in essentially any animals other than humans and chimpanzees and I think a woodchuck or some crazy big animal. So it's very difficult to find animal models. And the typical model in the field is something called a liver humanized mouse. And the idea is that this mouse is born with some sort of defect in its liver. And a common defect that people use is FRG. And so the mouse liver is sick, those hepatocytes are dying. And typically you rescue the mouse by injecting them with human hepatocytes. The background of this mouse is immune deficient, by the way. So it has the sick liver and an immune deficiency. So you can imagine it's a pretty unhealthy mouse. But the human hepatocytes will find the liver. You just inject them into the blood, but they find the liver and they grow there. And eventually you have a liver that is typically mostly human. And then you can infect it with hepatitis B and they'll grow. And so we wanted to ask, could we use a model like this to investigate a gene editing therapy for hepatitis B? So again, we'll use our Cas9. We'll have our two guides, 4 and 17, or control, just anti-GFB guides to see if anything happened. And so here's the experiment. Again, the paper we published in, there's the guides. Here's what we'll do. So this is the mouse model. And just to sort of cut to the chase with this, after we established a persistent infection for many months in these animals, we ultimately treated them with an AAV vector again, delivering the two guides and SA-Cas9, and then waited several weeks and then asked, have we done anything to the viral infection and so forth? So let's actually start maybe, oops, over here. So here's the animals. First, we put them on entecavir. We thought it would be good to stop the viral replication or slow it down as much as we could while this was happening. We got about a 3-log reduction with entecavir on that. Then we gave them our AAV therapy, waited a month. Some of the animals were evaluated at this time point. And then the rest we took off of entecavir and just asked, was there any difference between control and treated animals in viral rebound? And the answer, as you can see right here, is there really wasn't. So we wanted to look more carefully at these animals. And much like I showed you with the early HSV experiments, we definitely did have gene editing, but it was in the range of about 0.2% or so. So you can see some animals here, 0.3%. There's an animal over here, 0.02. So definitely gene editing of hepatitis B, but not at a level that would be thought to be beneficial. But then we thought, well, this is a double cutting. Maybe all we're measuring in this is the virus that's kind of left over. Maybe we've had a loss of these circular genomes for hepatitis B. And if we measure that both at the early or the late time points, this is just measuring the total amount of cccDNA per liver cell, essentially, you can see that in both our treated, in both the early and the late groups, treated animals do have smaller viral loads than untreated. Unfortunately, neither one of those trends met statistical significance. And the reason for that is that these liver humanized mice are incredibly expensive. They're very sick, as I mentioned. Typically, academic labs simply can't keep them alive. You end up working with a company who does this, and these mice cost, on retail, multiple thousands of dollars per mouse. We got some donated, used to us. They were actually quite old, had been used for other things, made the experiment even more difficult because now they're aged. But we didn't spend nearly that much money. But we really had no ability to go back and test and improve this. And the herpes work improved so much over time as we tried new things. How could we get this system into something where we could do it over and over and iteratively improve it? So, why have we had more success with HSV than hepatitis B? Well, maybe the viruses are just different. The biology is different. Could well be. Maybe the meganucleases are better than Cas9. You've talked about that. And hepatitis B is heavily chromatinized as well. There are differences in the AAVs we can use. You can use better AAVs with meganucleases because they're so small than you can with Cas9. But really, we wanted to just see whether we could improve all these things. So I mentioned, you have this $11 mouse for herpes versus this, $3,000 mouse for hepatitis B. Which disease is going to advance faster, right? So we looked at a lot of different models, and we've actually spent a lot of time, and I'll just kind of skip through these. But we actually settled on a model that was presented by a speaker right before me at the gene therapy meeting a couple of years ago, called the NSG Piz mouse. Now, the NSG is simply the immune suppression of the mouse. So it's a non-skid mouse. But Piz is a type of alpha-1 antitrypsin deficiency. So you may be familiar with this. There are children born with this. They typically have lung problems because the antitrypsin is important to prevent lung damage from trypsin. But that can be controlled now with medications. And what you see in people now is that there's also a liver pathology associated with that. So this model is really built to study the liver, but they found that you could humanize them, put the human liver cells into the mice, and they would engraft. And so the question came up, could we use this as a hepatitis B model? And one really nice thing about these mice is that even though they're sick, they're not nearly as sick as the previous models that had been used. They're actually pretty healthy. They're pretty easy to grow. You don't have to have a lot of specialized expertise. So we asked, in our hands, could we do this? So we got some of these mice. Again, they have the kind of a liver that's pretty sick. We give them another hepatotoxic agent. We typically either use anti-FAST, that's anti-CD95, or more typically now we use monoprotein. So an additional therapy damages their liver more, gives sort of room for human hepatocytes to grow. We put that in, and then we ask, could we challenge them with hepatitis B? So here they are engrafting over time really nicely. We measured human albumin over time, and you can see it goes up. We evaluated several human hepatocyte donors. And so they're definitely engrafting, just like had been previously reported. works very well. We found that monocrotaline typically worked better than anti-FAS. And here's what they look like. So you can actually see areas of human hepatocytes here interspersed between the mouse hepatocytes. So there's some mouse liver, there's some human liver. Human liver tends to be a little bit fatty. You can stain it with oil red O. But overall, they're engrafting pretty nicely. Here they are again. You can actually stain this with human cytokeratin and see that they are human cells. But importantly, for what we want to do, they do support replication of hepatitis B. So here we are. We can see if we infect them with hepatitis B, multiple animals, you can see that they do get infected, and they actually have really high serum HBV loads. These are very similar to human loads. So this model works really, really nicely. So there's replication throughout. So here we can look at hepatitis B surface antigen, and you can see that it really is in all of human hepatocytes. Human hepatocytes are stained in yellow. So if you overlay them, look at all the nice orange there. So it really works very, very well. There's just measurements of their CCC DNA and so forth. And you can see nice measurements of that. We have gone on to evaluate whether you could use these as a model for antiviral therapy. And the answer is yes, they work really well. So here is the viral loads growing over time in the animals. And then we treat them with entecavir or entecavir plus a capsid assembly modulator called CPX. And you can see, just like I showed you before in the very expensive model, you get a really rapid and several log decrease in viral loads with this. And then finally, for what we want to do, we would love to test gene therapies in these. So can you transduce them with an AAV vector? And the answer is absolutely yes. So here we're transducing with a vector expressing a green fluorescent protein. There's the human hepatocytes, and you can see all that nice green throughout them. They really, really work very nicely for that. So we are in the process of doing a gene editing experiment. I wish I could tell you how it's going to work, but we are taking some of our previous guides that we know have worked in the past. We're trying the same AAV we used before called LKO3, developed by Mark K, as well as a different one called MP59 that Nicole Pock developed. And now we're doing just more animals per group so that if that reduction in CCC DNA that we saw before actually was real, we will now have the statistical power to prove it. So we're really excited about this experiment in vivo. So I sort of mentioned what we're going to do. We're interested in looking at different Cas9 variants. We would love to have a meganuclease for this. We want to ask questions about what happens long-term if we treat them. And then very interested in non-viral delivery mechanisms like lipid nanoparticles and others, which are great for delivering gene editing, whether it's Cas9 or anything else. So I think just about the mice, they're actually a really powerful model. And we've actually had a lot of people approach us now to use these. So it's very exciting because they're just relatively inexpensive and very robust mice. These are the folks who did this. Rosanna, Michelle, and Dan led the hepatitis B work. Martina Aubert and her team lead the HSV work, and then lots of people and funders help. So with that, I will stop and thank you for your attention, and I'm certainly happy to answer any questions.
Well, it was extraordinary, actually. So we saw a lot of wonderful things. Actually, I will go directly for this question that is playing in my mind about latency. So, do you think it will be the same for HIV? I mean, that the fact that Cas9 will not be effective in latent form of HIV?
I think HIV might be a really good target for Cas9 because HIV literally integrates into the genome. So it is handled much more like a human. It is part of the human genome, basically, at that point. And we know that Cas9 works well against the human genome in general, that our genomes just don't get chromonized to the same level that these viruses do. So, and there are other groups who worked on this a lot. I mean, the group from Temple's worked a lot on this, and it seems to be working pretty well. So that's one virus where I think Cas9 may have a very good chance.
So again, in this same philosophy, so if we go for different disease, different like So we have here herpes and hepatitis B. So the strategy of the treatment, if I would say so, again, in the latency stuff. So should we program also when we have to do things? I mean, when we have to try to cure or have to, dependent on this latency stuff. You see what I mean?
Well, maybe. So if I answer, you can follow up if I don't answer your question. So timing may matter. What we observe with HSV is that if you have sort of a slightly inferior therapy that's really struggling to deal with the actual latent form, those latent circles, it can still be really good at destroying when that tries to replicate and come out. So when it makes new copies of virus, they haven't been chromatinized yet. They're brand new, and they're very easy targets. Cas9 can kill them really, really easily. So you could think about, having a therapy that's there and sort of acting in a more acute way. But again, I think for cure, those long-lived forms, we need to do something about them. So I think we do need something that can deal with the chromatin and that sort of form. Hepatitis B is interesting. Hepatitis B, I got caught. I wrote a grant years ago for hepatitis B the first time. I didn't really understand the virus, and some real workers in the field were really adamant. Hepatitis B is not latent; it's persistent. So, and it gets down to just the words I suppose you use, but it's never fully asleep like herpes is. So that circle's sitting there. It's very stable, but it is always kind of making some copies, and it is being slowly replenished. So that, it's an interesting issue. I mean, again, we thought we really wanted to completely break that replenishment with the antivirals, but we just couldn't knock it down to zero. We always had a couple logs, but there was still virus in the blood. So there, I don't know whether, just sort of a longer term therapy, it may be that we can target that, and it may be slightly easier than herpes, but because, we have hints of efficacy there with the Cas9. So this is the trouble. We just need to do more experiments, and we finally have the tools just now after 3 years of working on this, where we can actually start to answer those questions.
Of course. So maybe question in the audience. Oh, Laurie, we have here. She's running actually.
Oh, wow. Well, I'll wait. You don't have to run.
Yes, bye.
Hi, Dr.
Hello.
I have a question concerning the model, the humanized the NSG Piz model. So what is it that the Piz mutation is doing for improving the engraftment of the hepatocytes?
Yeah, so the PIZ model, so it is one type of alpha-1 antitrypsin deficiency. So there's different types. So here what happens is the alpha-1 antitrypsin has a structural defect that causes it to aggregate. So you actually get aggregates of the antitrypsin. So they get inclusions in the liver. So sometimes you can actually see inclusion bodies like you might with a viral infection or something, and it's just this accumulation. But that leads to the death, to slow ongoing death of mouse hepatocytes.
Or something like that?
Yeah. So basically, as the mouse hepatocytes are dying, you're giving very healthy human hepatocytes, and so they just kind of go, oh, I could live there. And so that's why you get these basically areas where the human cells have taken over, and then the surviving mouse hepatocytes. So their hepatocytes aren't as sick. I think that's kind of the trick, is that it's a less severe defect of the mouse hepatocytes than in the FRG, or another one's called UPA. And the mouse cells are just all dying, and so the mice are just barely alive. So I think, kind of having them less sick and taking our time allows us to have a healthier model that we can actually work with. Okay,
thank you very much. I have a second question concerning the non-viral delivery method that you intend to use for the potential treatment.
Yeah, so we've been looking a lot at lipid nanoparticles. So these are kind of what they sound like, little fat bodies that they're basically very similar to what's gone into a lot of the COVID vaccines for
Or
the
spread
mRNA
over the
vaccines.
site. Yeah.
Yeah. So they're pretty much that technology, but instead of a small, instead of delivering an mRNA to make a COVID thing, you're making a meganuclease or Cas9.
The nuclease enter into this kind of liposomes?
Yeah, so they can definitely be packaged. So we have data we haven't published yet, but I can just mention that if you use the LMPs in this humanized mouse model that I just showed you, the trouble that we found is that they go wonderfully to the mouse hepatocytes and they don't go into the human hepatocytes. And we don't know why. But of course, if we want to use them for hepatitis B, the hepatitis B is in the human hepatocytes. So we're kind of having a failure at this point. And so I think it's an artifact. So I went to the meetings and talked with the people working with the FRGs and the UPAs, and they've all actually seen the same thing, just nobody's written a paper about it yet. So I think, so it's an artifact of the models, I think, because we know that they can go to human livers or macaque livers, but they can't in these models, which is unfortunate because it's just going to hold back hepatitis B work with LMPs till we can figure that out. So we just I think we need a different LMP or something. So we're working with several groups to try to solve that.
Maybe with some kind of addressing receptors, you could go for something that is very different.
Yeah.
Extracellular
Exactly.
system or system
Yep.
like that. I have a very last question, and then I leave. Okay. What about the dose? So what kind of dose do you intend to use to treat HSV, for example, for when you go to a human body?
Yeah, gosh.
Again,
So I always have to, so I have to always translate this. So typically in a mouse with AAV, we're using about 3 times 10 to the 11th or so per mouse. So that, what does that come out to? I always have to do this in genomes per kilogram. We're about, let's put it this way. If you looked at like the approved AAV therapies like Zolgensma or something, we're about tenfold below that. So we have a lot of safety room, basically, in milligrams per kg. Yeah. So I think typically you see toxicity about 10 to the 14th there per kg, and we're about a log below that.
Okay. Thank you very much.
You're welcome.
By the way, thank you.
Good, great questions. Yeah.
Thank you. Merci, Logan. Any other question in the audience?
Sir, thank you for your speech.
Sure.
Obviously, most viruses take advantage of our weaknesses by cracking or hijacking our immunity. Wouldn't it be judicious to edit the human genes to fine-tune them in a way that improve our resistance and combat the infection? Thank you.
Yeah, that's a great question and a great strategy. So that's most advanced in HIV. Probably the farthest along gene editing approach is to target CCR5, which is the co-receptor for HIV, with the idea that if you could do that, the virus would just have no way to get in to the T cells. CCR5 isn't required in a mature T cell, so it's not even required through most of development. So it's a great target. So lots of groups have worked on that. We have worked on that. We've published some papers. It's not a complete cure, and we can't get all of it out of all the cells yet. And typically it's being done in a very expensive way where you take the cells out, you do it in the lab, you give the cells back 2 months later. It's not really practical to think about doing that for everybody who needs it. More and more groups are now thinking about ways to do that inside the body. And that's really exciting if we can do that, because it would be much less expensive, much easier to roll out to everybody. So that's a great example, and that's far along. Then people have thrown out ideas like targeting restriction factors and so forth. Those tend to be better known for HIV than for some of the other viruses. There's not an obvious target, and unfortunately, most viruses tend to use things that we don't have the luxury of destroying. We can't get rid. Herpes likes the just receptors on heparin sulfate. You can't get rid of that. So. But I think a smart approach to that and being aware as more things are learned about the molecular biology of these viruses, those targets may present themselves, in which case it's a great approach.
Yeah. Other question in the audience? Well, so then let me ask you, this might be a final question, because actually I'm dealing with drugs, etc. So I have still this confusion maybe. So is the fact that we will use like a DNA casseur, help me, people. Breaker, yeah. DNA breaker as a strategy. So if this will exclude any ethnicity difference. So if let's imagine that we have this CRISPR-Cas or whatever, so you will fight against Hepatitis B in Egypt and in the US, for example. We will find the same result, I mean, output, do you think, or?
Yeah, I think so. We have a paper on, for example, looking at Cas9 guides for all the different genotypes. So can you find a genotype that covers a guide that covers all the genotypes? I mean, biologically, I don't think there's any reason to think that the therapy in general would work differently, in different genotypes or different human genomic backgrounds. But the guides, you got to think about the guides, right? For sure. And we've done that. That's also an issue with HIV, if you want to do the same thing there. And even in diagnostics, we have a paper, we developed an assay to estimate the replication competent reservoir in HIV. And we were working on that for many years, and I guess we were slow, and we got scooped at this by one of the very best scientists in our field. But what we realized is that the assay that both of us were developing, and we published ours, were for B, which is what we have in the United States, where and a lot of the trials are happening, but actually isn't by any means the most common one in the world. And so when there were trials being done anywhere else in the world, this assay didn't work. So we developed one that's basically pan HIV that works in all the different genotypes. It was a lot of work and it was hard. But a lot of the things we had done, a lot of the software we had built to find guides were great when it was just a matter of making PCR primers and probes, because they're pretty much the same length. It's the same problem essentially. So yeah, and so we're well aware of that, and I think we're all over it, and I think it's going to be fine.
Okay, so I think people are happy after all this conference, and especially this one. Professor Keith, thank you very much for being with us.
My pleasure.
For giving us this wonderful talk. I wish you all a best day, and see you in another adventure and another occasion. Cheers. Bye bye.
Cheers.
Ciao, Misaki. Ciao, Misaki. Ciao.